It's a real pleasure to be here. I thank the graduate students for inviting me. It's always a pleasure to talk to bright young people, and you have a really good group here at Cornell. So we had a good discussion at lunch. As Fred mentioned, I'll be talking today about roots of the second green revolution. And see if we can get this to work. So here's where we were in the 1960s. If you fertilized rice or wheat, there's some more chairs in front if you folks want to come on up. Susan, don't be shy. You actually lost yield because the plants got tall and fell over, lodging. Now, Norman Borlaug and others developed dwarf varieties of rice and wheat incapable of lodging. Therefore, you could fertilize them and get a significant yield gain, like this. However, there was really no benefit to these new genotypes at low fertility. This was a combined package of fertility inputs and the right kind of genotype. So that was the historical green revolution. What I want to consider with you today is, can we develop genotypes that do this? So we have high yield potential, but better yield at low fertility. This was the benefit from the historical green revolution, one of the most significant agricultural advances of the last century, of course, as you know. Norm Borlaug received the Nobel Prize for this, passed away recently, saved the lives of millions of people with this technology. And then this area represents the potential benefit from a 21st century green revolution. That's what I want to talk with you about today. Well, first to summarize, I don't think this is really needed perhaps here at Cornell, but to summarize a few statistics about the need for a second green revolution. We have about 1 billion hungry people. Since I've been giving talks about our work, that number's been growing up. We started off saying there were 700 million hungry people, now it's 1 billion. Never before in the history of our species have we had 1 billion hungry people. It wasn't until about 1830 that we had 1 billion humans at all. So this is really a significant problem. And of course, that's growing over time. This translates into about 7 million children dying from hunger. It's estimated that over half of all childhood deaths result directly or indirectly from malnutrition in the developing world. And of course, the human population is continuing to expand. It's expected to reach 9 or 10 billion over the next several decades. Interestingly, serious estimates of the carrying capacity of the Earth are between 8 and 10 billion. So we're going to be close or beyond the theoretical carrying capacity of the Earth. And you know, in these developing countries, low input agriculture is what's practiced. In fact, according to the World Bank, this is the single largest human occupation. So if we should meet E.T. anytime soon, and they said, what do you do on Earth? We have low input agriculture. We have this hoe and we eat what we grow. That's the main thing we do. And in these systems, low input is low output. Water and nutrients are strongly limiting productivity in these systems. Here's some data. 3 important food security crops: maize, paddy rice, rice grown in water, and common bean. If we look at the top bar, that's yield potential under ideal conditions, what you can get in the field. So for maize, you can get 20 metric tons per hectare under ideal conditions. The bottom bar is what's obtained in developing countries in Africa. You're lucky to get a ton, so you have a 95% yield loss due to some type of stress, biotic, pests and diseases, or abiotic and environmental stress. Paddy rice, rice grown in water, you're still losing 80% of your yield. Common bean fixes its own nitrogen, you're still losing 90% of your yield. So certainly in these, even in rich nations, if you look at the USA, where obviously this year as an example, we have stress problems in the US, but in these developing countries, stress is causing very low crop yields, which is a primary pervasive cause of food insecurity. Now there's lots of particular proximate causes to food insecurity. Wars or disease or whatever, corruption. But you look over the planet, you look on a large scale, the fact is these people are just not getting the yields they should be getting from their crops. It's a big problem. This is one of the constraints we've looked at. Low phosphorus availability. This is a map we developed of soil phosphorus status compared to what crops need, where any red color represents suboptimal phosphorus availability. And you can see most of the planet is actually suboptimal phosphorus availability for plants. In fact, this is a primary constraint to life on Earth. Now, when we do that kind of analysis, we were using soil map data, which is based upon native soil fertility before people have used the soil. Most people in using soil make the soil worse. Soil degradation is a big problem. This is a coarse scale map from the United Nations showing regions of soil degradation. Red is very degraded. And here again, you can see even in the Mississippi Valley in the US, we have serious problems. Estimates are in Africa, as of 2006, 75% of the cropland was seriously degraded. And this, of course, is ongoing. Every year it's worse. Okay, so native soil fertility is a best case scenario. Actual soil fertility is significantly worse. Well, what can be done about this? Integrated soil fertility management would consist of using fertilizer inputs when they're available. We'll come back to that in a second. Soil management itself, which is very important. However, my experience traveling through many parts of the developing world is these techniques are very seldom practiced for whatever reason. Lack of information, lack of resources. People do not own the land, so they don't bother trying to conserve it. In fact, I live in an agricultural valley near Penn State. My neighbors are farmers. They don't practice very good soil conservation, despite several decades of investment by the US government in promoting soil conservation. So these are very important. I think they have limited impact on large scale currently. And then the third component would be getting the right plants out there, getting the right species and the right varieties of the species that have tolerance to these stressful conditions. And this is what I'll be talking about today. But before we go on to that, let's come back to fertilization. This is the most recent data available from the World Bank showing fertilizer use per hectare of arable land on a national basis on a log scale. And you can see here most of sub-Saharan Africa and significant parts of Asia are actually using negligible amounts of fertilizer. Every few years, the World Bank produces a new set of data, and we make a new map, and it looks worse than the previous one because energy costs are going up, fertilizer usage is going down. One of the countries we're working in, Mozambique here, in the past 10 years, has reduced fertilizer use by 90%. Okay? There are places here in Africa where fertilizer is 10 times more expensive than it is in New York or Pennsylvania, because they don't have fertilizer plants in these countries. Transport costs are significant, access is limited. So, you know, there are also questions about the long-term sustainability of intensive fertilization as a strategy. Certainly in the foreseeable future, in the near term, mid-term, it's hard to see how fertilizer is going to be a solution to these problems in these poor countries. Which brings us back to the need to develop adapted germplasm. How feasible is it? The first story I want to tell you is about common bean. It's one of the most important food legumes on Earth. We know it mostly as string beans, but also, you know, kidney beans, pinto beans, all those things are common beans, Phaseolus vulgaris. And in Latin America and Africa, as a grain, it's an important source of protein for people who can't afford animal protein, and it's also an important source of micronutrients for about 1 billion people. Here's a typical bean production system in the Andes, and it's kind of hard to see at this scale, but this is a maize bean polyculture, an ancient polyculture system growing in this village. And if you look at those plants closely, and you've taken my class in plant nutrition, or Leon's class in plant nutrition possibly, you'll know that this is a phosphorus deficient plant. I'm expecting it to lose 80 or 90% of its yield based on its appearance here. And in fact, world average yields for bean are only 5 or 10% of yield potential. In fact, we quantify this. We know where beans are grown in different regions, and then we can quantify the edaphic constraints, the soil-related constraints to productivity. We did a very conservative analysis where we only considered a region to have a stress if we were losing 75% or more of the potential yield because of that stress. Okay? This is a very conservative estimate. You can see phosphorus is by far the most pervasive problem for bean production. There are regions such as East Africa which have a wide array of problems, but phosphorus is certainly number one. Now, this is especially important for legumes. You need a lot of phosphorus, fix their own nitrogen. But I think if we did this for potatoes or any other subsistence crop, we'd see a similar sort of story with the addition of nitrogen as another stress. So here's how I started engaging this directly. I got my PhD in '87 from Davis. Went to work at the International Center for Tropical Agriculture, CIAT, headquarters in Colombia, South America, where for 20 years they had been doing this kind of brute force field screening, trying to find genotypes able to beat the check line. In this case, it was a Brazilian landrace carioca. And they had failed. It was a few months before I arrived there. They actually fired their soil scientist. The breeder had published a paper saying it was impossible to beat Carioca, and the whole effort was closed down. Now, it's worth pointing out that we're like 2 or 3 degrees north of the equator here. They can do 4 cycles of selection every year. So 20 years represents a lot of genotypes they looked at, and nothing was able to beat Carioca in the field when they look at yield. Now, I came into this with a physiology background, thinking this is, I think nowadays you'd use a different terminology perhaps, it's a phenotyping problem. What was their phenotype here? Yield, okay? What was their selection environment? A bad soil. It turns out they'd been driving tractors over this for 20 years and hadn't subsoiled it. It was hard, it was compacted. It had manganese problems, it had aluminum problems, it had low calcium, it had low everything. They just had neglected it, and they just did their screening, did their breeding for elite lines and then threw them in here to see how they did. And nothing beat Carioca, of course. This might have had good phosphorus acquisition, for example, but might have been sensitive to a disease that was there or some other problem, like aluminum. So I thought, no, this is not the way to attack this problem. We have to get scientific, just like the disease breeders and insect resistance breeders get scientific. You don't talk about disease resistance, that's meaningless. You challenge plants with specific isolates of particular pathogens and see how they perform, okay? So I thought, let's just put plants under just phosphorus. And let's look at performance in a more sophisticated way than just yield. And to make a long story short, we found that there were a number of root traits. We didn't start off thinking it was going to be root traits, but it turned out root traits were important for performance in low phosphorus soils. And good genotypes compared to bad genotypes had root traits that permitted them to explore the topsoil better. That's important because phosphorus does not move in soil very well, so it ends up being accumulated in the topsoil over time as plants die and leave their leaves on the top. So root traits that help you explore the topsoil are better for phosphorus acquisition. I'm not going to go through all these. I'm just going to give you a few examples to illustrate the kind of concepts we're talking about. Here's one of the first ones we observed, which is that the growth angles of this main axial root type in bean, which are called basal roots, varies. It can be steep like this one or shallow like this one here. Okay, this is from the field in Honduras. And when we cross parents of shallow and deep roots and get recombinant inbred lines, put them in the field, we see that the root shallowness, as you can see here, is closely related to phosphorus uptake. Large variation from about 0.5 units to about 3. That's like a 6-fold variation in phosphorus uptake associated with a variation for root shallowness. Of course, with these progeny recombinant inbred lines, we can also do genetic mapping. And so here's the 11 chromosomes of bean. Here in red are loci that are associated with yield under stressful conditions in the tropics, low phosphorus fields in the tropics. And the yellow, blue, and green are measures of root shallowness in a plastic bag of a young seedling in the laboratory. And you can see there are significant areas of co-segregation, like here in chromosome 11. Both of them are complex traits. There's a lot of different loci here. But the fact that we got significant co-segregation was promising information to us in terms of if we select for root shallowness in a plastic bag, we'll actually have something to do with performance in the field. Okay. Here's another trait we were working with. This one was not discovered by us. It's been known for some time. We're looking under a microscope at root hairs. So here's the result of scientific breeding. And this is an important line in Central America for many years. This is a landrace, okay, from a low phosphorus area in the Andes, never been touched by scientists. You can see it has longer and denser hairs. So once again, crossing these, getting recombinant inbred lines, we can look at the importance of this phenotype for performance in the field. Here's the field environment in the background in Costa Rica. It's a farmer's field that has weeds and nematodes and other things. And yet we have a very good correlation between root hair length and phosphorus uptake, about a 250% variation. Pretty nice relationship for field work, okay? So this obviously is an important trait for phosphorus acquisition. This is a relatively simple trait. 6 loci explained over half the variation. Now, of course, as you know, in breeding, you're trying to combine useful traits, and so we're interested in the synergism or in possible antagonism of these traits. So Magalhães Miguel, our colleague in Mozambique, recently did this study where we have in the same genetic background, in the same set of rels, variation for root hair phenotype as well as deeper shallow roots. So putting them in the field in Mozambique, and we have 3 rils for each phenotype class here. And we're looking at this short root hair, deep-rooted line as our baseline. Okay, that's like what we're comparing everything against. If we compare that with rills that have shallow roots, we see the rills with shallow roots in a low phosphorus field have 60% better growth. If we compare that baseline to long root hair rills, they have 90% better growth. So we'd expect if we had both root hairs, long root hairs and shallow roots, we'd have a 150% increase of growth, right? There's an additive effect. But what Magalhães actually observed was twice that. And that makes sense, really, because what we're saying is root hairs are more useful on shallow roots where they're in the topsoil than when they're on deep roots in the subsoil. Or, looking at it the other way, you could say shallow roots are better if they've got long root hairs on them. So this is just an example where we're looking at a number of these phene synergisms, phene antagonisms. I think they're very significant for these root traits, and it's one of the reasons we see such large phenotypic variability for root traits is the value of a trait very much depends upon the phenotype it's in. Okay? And we have to understand these things better for breeding. In this case, it's a win-win situation, and these two traits are being used by our breeding colleagues. Another thing we have to be concerned about, of course, is the world does not just consist of low phosphorus soils. Another major constraint in these production environments is drought. And unlike phosphorus, drought tends to be a situation where your resource is deep. Water tends to be deep in the soil in most drought situations. So here we're comparing, again, recombinant inbred lines that either deep or shallow, same genetic background. If we water them and fertilize them, they grow the same. Under low phosphorus, the shallow ones do better. Here's the environment in the background in Honduras. Under drought, the deep ones do better, as you might expect. So here's a kind of a challenge for breeders. So we don't want to obviously release lines that are drought sensitive and phosphorus efficient. That's not what we want to do. We want a line that's both drought tolerant and phosphorus efficient. So how are we going to do this if there's a trade-off? Now here's another reason I think that at least for these root traits we see such large phenotypic variability, and breeding for these traits is complicated. Not only are there phene interactions, but there's very strong trade-offs we have with different strategies. There's not one ideal strategy for root structure. That's why we see the large range of phenotypes we can observe. In this particular case, we're working on phenotypes that have both shallow and deep roots, kind of a dimorphic root system. I don't want to get into that today. Another possibility are traits that help us with both shallow and deep resources. Here's an example of one. One of the students noticed if you look closely at where these basal roots appear, some genotypes have only one position. Each position can make 4 roots. We're calling them whorls. This genotype has 3 positions, 3 whorls. So basically, this is from hundreds of wild and cultivated lines, the number of whorls is closely related to the number of basal roots. Every new whorl, you have 4 more basal roots. And the thing is, in this species, each whorl, each position has its own characteristic growth angle. So by having more positions, more whorls, if we go from 2 to 3, we hoped that we could have a broader range of soil exploration and maybe help us get both water and phosphorus. So here's Harini Rangarajan's work. She's using simulation tools to model this. I'm just showing you this to visualize what it looks like over time. So here's identical bean models that just vary in the number of whorls. You can see that this has a broader range of soil exploration. Now you can also see one of the trade-offs for this trait. I think you can see this, that if you have fewer basal roots, you have more resources to devote to lateral rooting. And these ones have less lateral rooting. So there's these kind of trade-offs we have to think about. But putting them out in the field, so here's one more work by Magalhães Miguel. Here's this red tropical soil environment, low phosphorus soil in Mozambique. And here we have rills, same genetic background, a set of lines with 2 whorls in blue or 3 whorls in red. And under low phosphorus, we've got a 60% growth in low phosphorus by having 3 whorls versus 2. So this trait helps with the low phosphorus. Katie Barlow did this work at our site in South Africa with drought stress, and she was looking at a population where we had 1 or 2 or 3 whorls in the same set of rills, and you can see they performed the same under irrigation. Under drought, the more whorls you had, significantly better growth. So this seems to be a trait that helps us with both shallow and deep soil exploration. And it's an example of maybe, this is being used in the breeding programs in Africa. Another possibility is to kind of do what African farmers normally do anyway. Instead of coming up with an ideal genotype that does everything, use a genetic mixture. In this case, we could have lines that are shallow and good for phosphorus, lines that are deep and good for drought, and then a mixture, a genetic mixture, that have both shallow and deep in the same field. Now, we thought this was a brilliant idea, and I shared this idea with Rebecca, and she said, oh, that's obvious. We give that as an example in plant breeding classes or something. But we thought it was our idea, and so the idea is you're using rills that look the same. The farmer doesn't have to be concerned about the fact that the seeds are different or anything. And at least in a market economy like in Latin America, you don't want a mixture of different seed types in the same field. So these look the same above ground. They're actually a multi-line. This is the work of Amelia Henry, who's now at Erie. And so Amelia did this work in small farms, on farm in Honduras. You can see a range of yield potentials going up to pretty good yield, down to more representative yields for Latin America. The multi-lines in red tended to generally outperform the single lines. After doing this, we talked to a statistician and found out we needed 5 times more sites to get, because these on-farm trials are noisy. So we're doing this more, and we're doing it with maize, but we think this is a promising approach, is a multi-line approach, instead of trying to get one ideal genotype. There's reduced competition between plants, and then whatever happens in that season, you've got plants that are doing the best thing. Another concern of sponsors was the system impacts. Of course, we're talking about root traits that assist in phosphorus acquisition. We're mining the soil. And the question was, are we going to eventually degrade the soil by mining phosphorus out of the soil? Good question. It really hadn't been approached from a research perspective. This was our hope. This was our model. If we look at a current bean production system, and they're usually grown in uplands on hillsides, the yellow numbers are kilograms of phosphorus per hectare per year. So in 1 metric ton of bean seed, which is twice the world average, there's 2 kilograms of phosphorus, okay, coming out into yield. Now, if we double yield to 2 tons, we're going to take out 4 kilograms of phosphorus in the seed. We're mining the soil. It's true. We're taking out more phosphorus than before. But the main loss of phosphorus in most of these systems currently, and we were using literature data when we first made this graph, you could be losing 100 kilos of elemental phosphorus per hectare per year in erosion, okay? So if you have a bigger plant, you'll be potentially reducing that loss from erosion. And just from the phosphorus perspective, without thinking of more food, more money, other good things, more nitrogen fixation, you could have a phosphorus benefit by increasing extraction of phosphorus. That was the idea. Fortunately, this is, once again, Amelia's work, working with small farmers in Costa Rica. So there's one of the farmers' fields we've rented from them. You can see how steep they are. Amelia installed these lysimeters made from local materials where we can catch the phosphorus running off. It's a 2-year production cycle with maize bean. You can see she was comparing deep-rooted versus shallow-rooted rills. You can see, I think here, the shallow-rooted lines are growing better in this low phosphorus soil. And Amelia found, in fact, these lines reduce phosphorus runoff 50 to 70%. So this is very promising. So in long term, you would think more yield is, in America anyway, more money, the farmer could buy fertilizers and everything is fine. Even short term, this increased extraction in the seed could actually be balanced by improved soil conservation. So here's genetic control of phosphorus cycling. So where's Carioca now? I can't even get the bean breeders at SIA to include it as a bad check anymore. It's so bad. We have stuff that's much better. Here's Steve Beebe, the leader of the bean program at SIA. This is some of his data. So here's Carioca with fertilizer, 2.5 tons. Without fertilizer, 700 kilos, which is the world average for Carioca. This is a reasonable level of stress. Some of these new lines with the better root traits, his is triple the yield of Carioca, over 2 tons without phosphorus. These are twice as much as Carioca without phosphorus. These have very good root traits. Steve has used these as parents in his selection program at SIAT, and those have been distributed through the SIAT bean network. So these traits have gone out into the Africa and Latin America. Although SIAT itself has not done trait selection or used the QTL we've identified. It's just we've screened parents and they've used the parents in their breeding programs. Now with support from the McKnight Foundation and other sponsors, we're trying to move ahead with this technology for Africa. working especially in Mozambique. This is Celestina Joshua, the bean breeder for the government of Mozambique. Magalhães Miguel is a crop physiologist working for Mozambique once again. Suarez Serinda is an agronomist. And so Celestina has been selecting for basal root whorl number and root hairs and developing a set of lines that are yielding better in these regional trials than the best material she had before. Suarez looked at the agroecological impacts of this. He's found significantly improved nitrogen fixation in these lines because phosphorus is a big limitation to nitrogen fixation. We're talking about 3, 4, 5-fold increases in nitrogen fixation. Less soil erosion, in fact, better benefits here than we saw in Latin America because of the soil type. He's shown that these new bean lines don't really disturb the maize that they're often grown with. And he has information that these new bean lines are better able to use locally available fertilizer sources such as low-grade rock phosphate, which they have in Mozambique. So this is a great situation. As a plant biologist, I can go home, right? We got lines with better yield, numerous ecological benefits, but of course the sponsors are not really interested in that. They're interested in people. And so collaborating with, especially with Jill Findice, a social scientist, used to be at Penn State, now she's at University of Missouri, Jill's tried to look at what are the potential impacts of these new seeds. Now she's done a number of ex-ante analysis trying to figure out what might happen before we make anything happen. Seeds have been released, and now she's in the process of figuring out sort of once we've released seed, what happens. Okay, so we're still in the process of this analysis. As part of the ex-ante analysis, Jill did a very extensive set of surveys going into several villages and having female interviewers interview female villagers, male interviewers interviewing males when the women were not present, so it's controlled and so on. And what she found is if we ask the people, like this is a Carioca seed type here, if we could improve the yield of that, what would happen? Okay, what is likely to happen? And women in particular were very happy about that because this particular grain type has no value in the local markets. It's not sold. So it's consumed directly by the household. So the women feed that to their children. This would be great because 70% of the households in Mozambique are food insecure. That's the kind of impact we would like. Now, if we ask them about this sugar type bean, it's a different situation. The sugar beans have market value, and marketing generally is handled by men. Not always, but generally handled by men in these communities. So the men will take that and market it. And what will they do with extra income? The most common thing they will do with extra money is buy beer. Now this is not, it sounds like, wow, what a waste, men, But it's more complicated than that, and we can't get into the details. Jill can tell you more about this. But many of these people do not own their own land. They get an allocation of land from the village headman, and by drinking beer with that person, maybe they're favored the following year. So there may be some family benefits from beer consumption in this case. There's a male agenda and a female agenda. Now we start talking about these kinds of high value. I have seen beans sold in Mozambique for $1.90 a pound. You guys in Ithaca are paying like $1 a pound or something like that, 75 cents a pound. These people earn 70 cents a day. So you can make a lot of money if you have access to like an urban market and have these seeds. You can make a lot of money. What are you going to do with that lot of money? And the women get very concerned when we start talking about this because this is a polygamist environment, and he might set up wife number 5 or 6. So he's optimizing his reproductive output by having lots of wives and children. And she's interested in feeding the kids she already has. So it's very interesting, and Jill has seen evidence of sabotage, where it's like the women will not, encourage the growth of lines that result in more wives and stuff. It's a very interesting situation, very complex situation. It's not solved by having better yield. There's all kinds of other things that happen. So Jill has done this kind of very fascinating network analysis. This is a very small part of her data set. She knows physically where each household is located in these villages, who talks to whom, how they share seeds, how they share information about seeds, lots of interesting information there. This is just like a very small sample of the kind of things she's pulling out. If we look at 4 households and we look at how the women and men exchange information and exchange bean seeds or information about bean seeds, you can see like in this example, the men have a more extensive, maybe each man interacts with fewer other men, but the networks are more extensive because they travel, they trade, they drink beer. The women tend to be more tied to the household where they have children and farming to take care of. So what that means is if Celestina would introduce a seed that the women really like, there's no formal seed system in these communities. It's sort of person to person. That new seed is going to have a hard time diffusing. If she introduces a seed that results in more beer and more wives, that information, that seed might go quickly, okay? So, so this is something that Jill has thought about, and how can we work with the existing seed system to promote outcomes that we find desirable? And we don't have time to get into it now, but it's just to illustrate some of the complexities of how this works. Now, that's the story with bean. And the McKnight Foundation some years ago asked us if we could do something like that in South China. This project was led by my friend and former student Xiaolong Yan, who tragically passed away from cancer a couple of years ago at South China Agricultural University. And the idea was to see, because bean and soybean are similar in many ways, and so this is what commercial soybean look like. And we were thinking, well, maybe if we brought in these shallow rooted lines, this would be good for these low phosphorus soils you find in South China, where soybean is a food security issue. And I think, Rebecca, as a good scientist, was very skeptical about this whole thing. I can still remember the time they had already funded us. And Rebecca was in the field in South China, and we dug up some plants and showed her the shallow roots. And it wasn't until that moment that she really thought, oh, maybe these guys are for real. She wasn't quite sure, even though she'd already funded. But so this was the idea, and Xiaolong's team, including breeder and so on, a large team of scientists there were selecting based upon phenotypic selection, using growth pouches and other methods, phenotypic selection, and then taking those into the field, have been very successful. The current leader of the lab, Hong Miao, she's been here at Cornell working with Leon. Xiaolong's assistant is now head of the lab. She tells me that they now have 15 of these new pea-efficient shallow-rooted lines released in South China. They're doing very well in regional trials. Here's an example of some of the differences. And so this is not something that was just useful in bean. This approach of selecting for shallow roots, in both cases in bean and soybean, it's phenotypic selection. People are not using the markers we've identified, have been successful. Now I want to tell you a story about corn. And for phosphorus, we found the same kind of traits and the same similar kind of genetic patterns for phosphorus efficiency in corn as in these legumes. It's not a legume-specific thing. But of course, the real interest in corn, I think, is water and nitrogen. You guys know about corn. It's a huge plant. It needs a lot of water and nitrogen. It turns out nitrogen is not only a big limitation to food production in the tropics, but in the US, the biggest cost of growing corn is nitrogen fertilizer. The biggest environmental cost of growing corn is nitrogen fertilizer. The biggest energy cost of growing corn in the US is nitrogen fertilizer. Estimates are 2/3 of the nitrogen fertilizer we apply in the US is never taken up by the crop. It goes right into the environment and causing pollution. So it seemed like that's an opportunity. If we could go from losing 60% of the nitrogen to even 50%, that would be huge. That would be hundreds of millions of dollars a year for US corn growers and big environmental benefit and better food production in the tropics. So nitrogen is very different than phosphorus. It moves. Here's a simulation of parameterized in the Pennsylvania soil. The red is nitrate. In the spring, you have rainfall events, which then push. This is actually actual rainfall events and actual nitrate pulses that we measured. This is put into a computer model we have. You can see how the nitrate is pushed down into the soil every time it rains. So it's kind of a race. These roots are trying to grow into cold soil and catch that stuff before it is lost. This is one of the biggest losses, if not the biggest loss in most systems of nitrogen. It just leaches out before the plants can get it. So we have an idiotype. Sort of our idiotype for phosphorus was this topsoil foraging. Idiotype for nitrogen and under drought water, it's a similar resource, a deep resource, is what I'm calling steep, cheap, and deep. Steep being a combination of architectural traits, cheap being anatomical traits that allow us to get deep roots quick enough to catch that nitrate before you lose it. And then if you have deep roots, you also have drought tolerance in most environments. Now there's a lot of things here. It's a lot of hypotheses that are mostly untested or unpublished. Just to keep it simple, I'm going to focus on just a couple of traits to give you an example. I'm going to talk about these two traits right here. One is a anatomical trait, RCA, root cortical parenchyma, and one is an architectural trait. So this root cortical parenchyma is, you're probably familiar because of Susan's work, and many of you here are working on rice. It's been researched a lot in rice. This air space, this is living cortex through programmed cell death is turned into these air spaces. Very important in a flooded soil like paddy rice to get oxygen down into the roots. Now, it's been known since 1980. This we're seeing here is natural genetic variation of unstressed plants from the same real population. But it's been known since 1980 that nitrogen stress, phosphorus stress, and now we know sulfur stress and drought stress, and we know high temperature, can all induce parenchyma formation. Now, these things have nothing to do with oxygen availability. Why are plants making parenchyma in response to all these soil stresses? We thought several years ago that it was because it made them cheaper. Instead of having this living tissue that's full of phosphorus and doing all this respiration, we have air spaces which don't require phosphorus or nitrogen or sugar to maintain. So you make a cheaper root. In fact, this is, percent parenchyma as a cross-sectional area in maize, and here's respiration. You can see you can cut respiration in half by forming parenchyma. So it's going to be a significant effect. This is a model of how parenchyma forms in maize roots. It's developmentally regulated. It's a complex pattern, but we've mapped this, and we know generally how this happens. The hotter colors is more parenchyma, okay? And so and then we know how parenchyma affects respiration and nutrient content and stuff. So using this kind of modeling approach, we can begin to ask, we can connect it to our nitrogen models and phosphorus models and see if we had parenchyma, would it help us with nutrient capture? Would it make enough difference to matter? Okay, here you can see, in this movie, you can see the blue streaks is where the roots have taken up the nitrate, okay? So here's, I don't know if you can see this with the lights being on. Can you guys see that? It's, so here's two maize roots, identical except for the presence of parenchyma. They're growing in low phosphorus cyberspace. There's a shoot here that you can't see that's doing photosynthesis and stuff. And so what's happening here, the difference is this ends up growing more with the parenchyma because each section of root is cheaper. That means the plant can grow more roots, get more phosphorus, and have more photosynthesis and so on. So the models were telling us, yeah, that effect actually could be significant. In fact, this is Jauke Postma's work, who's now in Germany. He's looking at for nitrogen, for phosphorus or potassium, as stress increases and as the grow rate of the plant decreases as a result of stress, the value of parenchyma increases to very significant levels. Now, our modeling here is only 42-day-old plants, which are already taxing the Penn State supercomputer cluster at 42 days. We could take it longer, but it'd be kind of a problem. So even in fairly young plants, we're seeing 60% benefit. We're also seeing that even under optimal conditions like in New York, having parenchyma could reduce your requirements for nutrients. A 13% reduction of nitrogen requirement for maize, as I mentioned, is like over $300 million a year for US corn growers and a big environmental benefit. So this was interesting, but I didn't really believe it. How could it be that important? So we've of course done field work. This is work of Patampong Sangwalai, a Thai government scholar, at our site in South Africa where we have low nitrogen fields comparing high and low parenchyma maize rills. These are inbreds. And he's shown in greenhouse and in the field that if you look at rooting down here deep in the profile, you'll see significant benefits of having deeper roots in these high parenchyma lines. Here's some biomass data. Under high nitrogen, they grow the same. There's a set of rills that are high and a set of rills that are low. Double the biomass at flowering of the high parenchyma lines. This is very promising. Indicates that maybe the model wasn't wrong. The value of parenchyma is also supported by this work by Jinming Zhu, published a couple years ago. We're using rainout shelters here in Pennsylvania to create drought conditions, and Jinming was using high and low RCA lines from the same rill population. Here we're in the field. So here we're looking at deep rooting, low RCA lines, high RCA lines. Red is water stress, blue is well watered. And you can see the high RCA lines have much deeper roots, especially under water stress. The high RCA lines have better midday leaf water status under drought. And in terms of yield, the high RCA lines under drought have 8 times better yield. So here's what the yield looked like, the water stressed on top, well watered on the bottom. So we cut off water entirely with the rainout shelter when these plants were about this high. There's a clay soil that holds water, but this was a pretty serious stress. You can see some of these low parenchyma lines really suffered. look at this one here, it's pretty decent considering it didn't get any water for most of the season. So this is very promising to us in terms of the value of having these traits that give you cheaper roots, anatomical traits for cheaper roots. Now, there's other traits that haven't been looked at. Now, if I read German, I would probably go back into the 1860s literature and see if some German botanists had talked about these things. I'm not aware of more recent research on these things. Natural genetic variation for a number of anatomical traits is interesting. This one is the number of cortical cells in the cortex. So we have a 5-fold variation in that parameter. Here you can see one with relatively fewer and relatively more. And that is directly related to root respiration. You can half root respiration by making the cortex thinner. And this is cell size. Bigger cells are cheaper because they're mostly vacuole. So no nitrogen necessarily, no phosphorus, no mitochondria, mostly just space, just water. So we have 10-fold variation in cell size. Once again, associated with 2x variation in respiration. So I'm excited because I think there's a number of anatomical traits that have not been exploited that can give us cheaper roots. That from the Arenkema story, you think these might be useful. So I wanted to mention briefly architecture with steep, cheap, and deep. Steep, this is the work of Sam Traxel, who's now at CIMMYT. This is the NAM 5000 population I'm sure you're familiar with from Ed Buckler's work. We've actually phenotyped this 3 times in the field, Ed, and we're going for number 4, and it's a ton of work, so I hope it works out. But just in terms of the phenotypic variation, we're looking at variation in crown root growth angles. This is a relatively shallow one, relatively steep one. The variation goes from 20 degrees from horizontal to 90 degrees, almost straight down, okay? And that variation in angle is associated with depth. We measure depth by the D95, the soil depth at which you have 95% of your root biomass, measured from soil cores. There's a good correlation between D95 and that angle. So when you measure the angle, you're actually getting an estimate of root depth. And then comparing a set of shallow and steep lines, Sam found that the steep lines had better water status, and here a 3.6-fold yield increase in the field under water stress in rainout shelter. So this is a promising trait also. And we have a number of architectural traits that we think are interesting. This root angles is also associated with nitrogen capture, both high and low N. We have correlations between yield and these root angles. So we're excited about this. Now for phenotyping, as you know, this is the problem. You talk to breeders about, let's breed for root traits. Okay, how are you going to do that? That sounds like a lot of work, and you kill the plant and stuff. So how do you do this? So we developed a technique. Our main one is called Omics. The essential tool is a dedicated graduate student and a shovel. This is Jimmy Burridge. He's one of our bean students, and you basically just excavate the roots and look at them. It's about 2 minutes per genotype in clay soils or sandy soils. And we're going through about 10,000 lines a year currently on a variety of crops. And you just basically, we started off visually scoring. We're now moving more towards automated image analysis type systems. We take a picture of everything before we throw it away. This is not a Hollywood type picture. This was just a random photo from the thousands we've taken. You can see little bits of mud on there. And you're basically scoring the root crown. So you can look at the number of things, the angles of things, the branching of things, the diameter of things. Anything you can see, we score. So let me give you an example of brace root angles. This is different than crown root angles I showed you before. Brace roots are coming up above the soil line. So you can see these brace root angles are shallow or steep. Once again, very large phenotypic variation in the NAM 5000. And when you do this QTL analysis, you can find lots of interesting markers. Here in this case, for this trait, 42 QTL, some of which seem to be quite significant. Of course, if you put this through the genome-wide analysis, it radically simplifies. Many of those QTL disappear, but we still have interesting hits. Here's this trait, and we see a locus here that seems to be interesting. So we're working with Sean Kepler to try to track some of these down. And for the anatomical traits, we were hand sectioning and then putting them through a software we developed called RootScan to automate the analysis of these anatomical traits. We can pull out 15 or 20 anatomical traits from these cross sections. Here's a Arenkema in red, and here's xylem vessel diameter or other things you can measure up there. And this is here, for example, variation in the IBM for these anatomical traits. A lot of variation. Here we have a lot of QTL identified. So this will be interesting also. Now the problem was hand sectioning. Okay, so this is a new technique developed by this Ben Hall, an undergraduate, 7th or 8th year engineering undergraduate, a brilliant guy, but too smart to get past classes and stuff. So we came up with this method. It's Penn State is patenting it, and I'm not supposed to tell you how we did it. I'm sorry. But basically, that was a real time. I can go through it again. This is a real time. This is what we do. And you can see these traits. So instead of doing hand sectioning, which you can do like 5 an hour, this takes like less than a minute to get all this information. And an undergrad can do it. And so then you end up with a stack of images like this, where you can see a number of traits in there. And then you can do image analysis. This is like a minute's worth of imaging here. Ben has pulled out a quarter section so you can see. We're going to go through a xylem vessel. He picked up like bugs from the floor and did bugs and a fried chicken bone and stuff. You can do. So here, the yellow is going to be the Arenchyma lacunae that we're trying to measure. So working with colleagues in Germany, we're figuring out how to do 3-dimensional analysis of these volumes. But really good tool for these anatomical traits, which has been bottleneck, has been the hand sectioning that we've been doing. So that's exciting. And then to be able to see how, remember how phene synergism might be important, to be able to see how these anatomical traits might interact with architectural traits. Here's our SimRoot model that Jouke Postma developed with this architectural model with the heat map in the red being the more Arenchyma. And then Jia Wu from Beijing, China Agricultural University, has helped us develop an anatomical model. So now we have a spatially explicit model of every root cell in the root tissue. This is based on actual measurements. And here, in fact, you can see the vacuole, which is important, and cytoplasm. So then we can ask questions about how changing a trait is likely to affect architecture and whole plant resource capture in silico, which is nice because we're working with like, I don't know, now it's over 20 traits in our lab. So the possible interactions among 20 traits is very large. Having these computing tools is really important to help us hone in on the most interesting scenarios before we do field work. So for example, here's some work by Eric Nord and Larry York looking at this phene synergism between architectural and anatomical phenes. Here we have Arenchyma, and here we have number of crown roots. This one, it's an interesting trait. It varies in maize between like, 6 to like 50. Huge variation for this trait. It's got to be significant. So we're interested in that trait. Here's an architectural trait, an anatomical trait. Here's a performance in terms of root length at low nitrogen, at high nitrogen. Now the point here is that these are not simple, straight, flat planes. There's complex interactions, which we would expect. Arenchyma, which makes roots cheaper, is going to be more beneficial if you have a lot of roots than if you have a small number of roots. So we're using these modeling tools to help kind of guide our thinking about what integrated phenotypes are most promising for field exploration. Let me take the opportunity, since I was invited by graduate students, I can take a little kind of professorial role here and give you a little sermon or perspective in this one slide about something I think. It came up in our lunch meeting, which was good. And that, there's obviously a huge interest now in this very fascinating, amazing stuff that we're learning about the genome. There are these huge pressing issues we have to tackle at the system level, okay? And in our work with these root traits, and I think in general, I think I just messed up your microphone. You don't know, Jing, if you can hear me okay, do you? I'll just hold it here. It's been very useful to know what's happening at the phenome level. What are these phenes? What are their trade-offs? How do they interact with other phenes and with the environment? How do integrated phenotypes perform? I mean, and of course, this is how evolution has occurred based on phenotypic selection. This is how crop domestication and traditional crop breeding has occurred. This is how our colleagues are doing in China and Africa and Latin America, are doing bean breeding for root traits right now, phenotypic selection. So, I mean, we can't forget that. And there's a lot of things, certainly in the root phenome, but I think in general, in the plant phenome, this is kind of a bottleneck. I interact with colleagues in the private sector who have told me, Jonathan, we can get in terms of the genome information, we could get a corn plant get up and dance, But we don't know what we want it to do to be nitrogen efficient or drought. What do we want it to do? That's kind of an organismic, or it's a question that integrates things at a different level. that I believe is kind of a bottleneck if you look at what's being funded in Washington, what's being published in the top journals. This is not getting as much emphasis as this, right? And I just suggest to you as future plant breeders that you not forget this part. It's important to know about this part, okay? I don't know what the right balance should be, but I think that currently the balance is too much on that side. Okay, so what's the summary here? Root architecture is an important component of soil resource acquisition. Topsoil foraging enhances phosphorus acquisition. Subsoil foraging enhances nitrogen and water acquisition when you have leaching, which is most of the time. Several anatomical and architectural features affect the metabolic cost of soil exploration, which is an important component of soil foraging efficiency. Bean and soybean genotypes incorporating root feeds for superior phosphorus acquisition are being deployed today in Africa, Asia, and Latin America. The steep, cheap, and deep idiotype for nitrogen and water acquisition appears promising. We're working with breeders in Malawi. We haven't had field impact yet, but it appears promising. High throughput phenotyping platforms are available. Numerous genetic leads are identified. Prospects for the second green revolution. Breeding for yield at low fertility has been successful. In the few cases it has been attempted. I have to say, this topic is met with a lot of skepticism. Maybe it's just when I talk about it, they're skeptical about me. I don't know. But people think, yeah, can you really get plants that need less water and nutrients? Yeah, you really can. Bob Schaffert, colleague in Brazil, likes to point out that animal breeders have developed chickens and pigs that need a lot less feed. The feed to meat ratio has gotten a lot better because of breeding. We can do that with plants. The few times it's been tried, it's been successful. And I have to hand it to the McKnight Foundation. At that time they started the work in China, it was sort of a new and different thing, and it worked out, but they took the risk. I think more people are beginning to realize this might be possible. Aluminum, you all know from Leon Kochan's work here at Cornell, aluminum tolerance is working. It's being deployed in places like Brazil today. It's had a big impact in world agriculture. Phosphorus is more complex than aluminum, but it is working. Nitrogen, quite complex, but it's promising. People are using manganese tolerance in breeding. Potassium, calcium, magnesium. If I was a young student wanting to take on an important problem that is neglected, I'm not aware of anybody breeding for these traits, which are important for acid soils in the tropics. Leon, are you aware of anybody breeding for these traits? They're neglected. But we should be able to make progress with these traits. Recent advances in root biology are making it feasible for the first time, really, to design root systems. We could say, if you're a corn breeder, in Iowa, here's the kind of environment I'm dealing with. In Georgia, it's different. In Malawi, it's different. We can kind of design root systems for those environments. The bottleneck, I believe, although clearly there's more to be done at the genomic level, is the phenome. What do we want the plants to do? I think that's the bottleneck. We need more information about the long-term system impacts, but they should be benign. Note that there's no resistance breakdown with abiotic stress. Once you have a drought-tolerant line, it's always going to be drought-tolerant. It's not scale positive, which is economic jargon for you're not just benefiting the wealthy farmers like the first green revolution did, because like a nitrogen-efficient crop is going to benefit New York farmers, it's going to benefit African farmers, okay? And these lines would have reduced pollution in rich countries, which is increasingly a concern, especially in Europe. Well, it's a concern in China. I don't know to what extent their policy is reflecting that, but yeah. As soils degrade, fertilizer becomes more expensive, and the world population expands. Certainly the need for this is only going to grow in the future. Now, I've obviously been summarizing the work of a lot of people. This is our current lab configuration. Thank you very much. Fred, are you moderating, or should I do this? I can do it, yeah.
So looking at Rankema, since they're induced under stress, I think there's some sort of cost to having them. They aren't just always beneficial. What's the cost?
That's a very good question. I think there are costs to all these things, and we have to understand them. And so that's a good question. We're trying to understand that. Some possibilities. We haven't published yet, we've shown that when you have a lot of Rankema, it's hard for water and nutrients to get across the root. But now Rankema is developmentally regulated, so the young parts of the root don't have it. So the plants may be able to tolerate that, but there's reduced transport of water and nutrients across the cortex. There's no place for mycorrhizas to live. That's something we're looking at. I was talking with Rebecca this morning. We're concerned that maybe these higher Rankema roots, a fungal pathogen can enter and then have an easier time moving around in the root. You know, we do field work. We don't necessarily see that, but there could be situations where that's a problem. So we're trying to understand, we're interested in the physical properties of the roots. Are they more sensitive to compression or compaction if they have parenchyma? And so we're trying to understand this. We're trying to kind of understand the fitness landscape for these traits. And it's complicated, and unfortunately there's not a lot of people doing this kind of work. So we don't know the answer to that question. But I think we should, you're right. When I see variation, I look out and I don't see anybody with 3 eyes. I mean, 2 eyes is the way to do it. But you look at roots, you see all these phenotypes. It's like, that means there's, different solutions for different environments, or the benefits may vary, or, and that's what we're trying to get a grip on. I don't think it's going to be easy. Yeah.
So I've always been skeptical of idiotype breeding, but your talk is sort of convincing me that I've always been wrong.
Thank you.
I don't understand why I've been wrong. Sort of, if you want high yield, it's a good idea to breed for high yield. Why do you bother with shallow roots? Why does that work better?
Yeah. So why did they fail at CIAT? They had a complex selection environment that had multiple stresses.
I mean, they could have done it right.
They could have put it just under phosphorus stress. But in that particular crop, also corn, which we work with, also soybean, it's like the performance of this line will depend upon its latitude, its elevation, the photoperiod, all kinds of factors. It's expensive and laborious to do multi-location yield trials. There's a lot of noise. Monsanto can do it. CIMMYT could do it with large resources from the Gates Foundation. But most of the time, yield is a pretty noisy, it's an important parameter, clearly, and local breeding has to look at that. But in terms of more strategic breeding or pre-breeding support that we can offer from research, I don't think yield is a good trait. Yield is a combination of everything the plant's doing. We don't do it for disease screening. If we're looking just in disease tolerance, you don't just throw plants out and see what yields better. You try to get more specific and say, how do my leaves react to this inoculant, And I think it's exactly the same thing with abiotic stress. Why would it be different? Yield is, if I can say, I've given you the roots to get the water, now you guys in your practical breeding put in your local adaptation, your local disease resistance, you should get better yield, right? That doesn't mean in my good root line I have all the other things I need to get good yield. You know what I'm saying? It's like you're making it so hard. It's like maybe there's 10% of your lines that have good root hairs, and maybe 10% of them have good root angles, and maybe, before you know it, you have to deal with thousands of lines to see a good one, which gets lost in the background variation. I don't know. I mean, you look around the world, and where yield screening for stress has been successful, it's been large, integrated, well-funded programs, usually with large uniform environments like Cerrados in Brazil. And you try to map that onto like Eastern Africa. They might have goats, and they might have polyculture. I mean, I don't think it's the most cost-effective way to go. I think it's trait selection. I'm not a breeder, though, so you guys tell me if I'm wrong.
Yeah. To jump in there. There might be 2 different types of breeding. John Luke, I presume in the U.S. where we have plenty of inputs and trying to figure out how to increase yields over what's already pretty high. But when you have aluminum tolerance, when they bred specifically for phenotypic aluminum tolerance, has been very successful. When you have that specific abiotic stress or combination, you need to overcome them. And I think I'm not a breeder, but it sounds like from the work I've done in this case, that we can do this type of breeding for specific abiotic stresses. In your world, where particularly in the U.S., where and I'm just assuming we don't necessarily, because we have the money to put in the inputs, we're dealing with trying to raise the bar without aluminum toxicity and phosphorus emissions.
The counter argument is you have these 20 phenotypes, and you're wondering about their interactions, and there's 20 squared different ways of going wrong, right? And if you just go for yield, there's only one way to go.
Yeah, and you'll make incremental progress. It's laborious and noisy, like I say. I think another factor is that we have found another problem at CIAT I didn't mention was these guys were thinking about disease. All breeders, were trained to do disease breeding. So they're all breeding for virus and disease and maybe some insect resistance and yield, potential. And then, oh, by the way, let's put them in this bad soil. Well, you look at the roots, they're terrible. They'd already been pre-selected against all the land races that had good root traits, because the land races didn't have other good agronomic traits. By looking at the phenotype, we can go into the land races and exotic stuff and bring that in where we might have lost it because of high input pre-selection, So I think it's been very informative. Even like we work with Juan Carlos Rosas, most important bean breeder in Central America. he has a recurrent selection program where he's looking at, I don't know, 12, 13 different targets of interest, right? We took his like 300 breeding lines and phenotyped all of them for root traits. And here, now you have the data, Juan Carlos, you know you're going to get good root hairs from here and this from over there. And he's not necessarily looking at the roots anymore, but he knows the sources, he knows the parents, you know. I think that's been helpful to him to kind of guide. It's like an informed, intelligent breeding strategy rather than, gosh, it works, I wonder why. Probably has good roots. That's where they were for years. They didn't make any progress at CF. That was, I mean, that's the problem. And Simmet was able to do this because they had massive funding, they had massive numbers of sites, brute force selection. They still don't know why they have drought tolerance and nitrogen efficient lines. They don't know, which means we could improve it further perhaps if we brought in the right traits, right? You can stack these things up. That's another reason to do it. If four things all combine synergistically, what's the chance you're going to get all four in a good adapted line? Pretty small. But if you can find the four independently and put them together, you might have a chance for a quantum gain. Like you saw a 300% increase in growth in the field of beans. You know. But I could be wrong. I'm not a breeder, so. Yeah.
Perhaps this is simplistic, but when you look at native bean germplasm, there's the determinate and indeterminates. And the determinates are much more fibrous root system, indeterminates much more taproot, kind of mimics the upper leaf architecture.
No, I don't know if that's true. We wrote a paper about that years ago. Okay. I don't think there's a root shoot homology.
Okay. Do you see that at all in your selection for different roots? Is there anything above ground that correlates with the different root systems?
Unfortunately not. Breeders would love to be able to look at the shoots and say something about the roots. And we've specifically looked at that for that and never seen it. So we haven't seen that yet.
Jonathan, I think that's all the time.
That's all the time. I'm sorry. Thank you very much.